Everybody, and welcome. My name is Charles Nicholls, and I am a co-founder and trustee of the Carbon Community. I'm delighted this evening to be joined by a truly megastar panel of environmental scientists who've been working with us on this landmark study. We're calling it a carbon study, and the event is a carbon study in conversation. So this is really a conversational style event to look at really what we're doing, why we're doing it, why it's important, and you know, what we might expect to come out of this. Let me just very briefly introduce everybody, and if each of you could just sort of wave as I go round. I'm going to do it in alphabetical order. So Dr. Colin Averill of the Crowther Lab at University of ETH Zürich, Professor David Beerling of the Leverhulme Centre for Climate Change Mitigation at the University of Sheffield, Professor Martin Bidartondo, Imperial College and London and the Botanic Gardens Kew, Professor Pete Smith, Climate Exchange and University of Aberdeen, Dr. Laura Martinez-Suz of Royal Botanic Gardens Kew, and Senior Lecturer Bonnie Waring at the Grantham Institute on Climate Change and the Environment at Imperial College London. Well, thank you all to our panelists for joining, and thank you to all the audience for coming on to listen and learn a little bit more. To kick off, I'm just going to give a brief introduction into what we're doing and to give you an overview, and then we're going to dive right into it with the panel and get stuck into the science. So let me just give just a couple of words really on the Carbon Community. The Carbon Community is a new charity which has been formed in about the last 2 years, and it's really dedicated to creating forests and accelerating carbon removal with breakthrough science. If we look at the environmental challenges that the world is faced with, there is too much carbon dioxide already in the air, and therefore carbon removal is the process of drawing it down from the air and storing it in the ground, in this case in trees and soil. We think about our mission really as being accelerating natural processes, so you can call those nature-based solutions, and we're going to be talking about two nature-based solutions that we're deploying in conjunction with new forest creation at scale really for the first time. And so what we're doing is we're really blending trees and science and people, and ultimately connecting people with nature is a core part of our purpose. We have a new site, a flagship site called the Glandir Forest at Kinghordy in Wales, just outside Llandovery. And this is our first project. It's primarily a broadleaf forest site of about 170 acres and more than 100,000 trees on the site. Nestled within the Glandir forest is our world first carbon sequestration study, which is really what we're going to be talking about today. Now, this is a field trial at scale, and it's combining two nature-based solutions not previously deployed together at this kind of scale. The photograph here, you can see back in November, some of our test cells that we're beginning to mark out. And each of these test cells allows us to test different nature-based treatments and understand their effects by comparing them against each other. We're really trying to enhance and accelerate carbon sequestration, and this is both a story about trees and a story about soil. And much of the data that you can easily find on, to answer the question, which is if I plant a tree, how much carbon does it capture, is really focused on what you can see above ground. It's the bit that you can see easily and measure. And yet we know that if you look at an ancient forest, 75% or more of the carbon is actually stored below ground in the soil and in the roots. So we need to look at both together really to make that make sense. And this is the key part of this story. And obviously what happens below ground is really tied up with biodiversity. So there is a biodiversity element to this, which is going to come through as we talk about it. Now, we mentioned that we have a series of test cells in effect, what we're doing is we're taking 8 different treatments and a control. And the control allows us to then benchmark what would have happened if we hadn't done anything at all. So the 8 treatments are replicated 8 times. And this is a test grid which was designed by Colin originally. And in effect, what it does is by using this amount of replication, it gives us real statistical confidence in the results, because in the reality of fields on the ground, you end up with little microclimates, and some fields are south facing and some are southwest and some are southeast, and there's all these differences in the amount of water and all this sort of stuff. So by doing that replication, we get great confidence in terms of what we're doing. It also means that what we're doing is quite significant in terms of scale. In the control groups, you can see here we have a couple of patches of bare field, if you like, where there's no planting going on. And these are controls, and these are allocated randomly within the groups of 8. In some cases, they're on the edge of the group of 8 or group of 9, including the control. In some cases, they're on their own, and in some cases, they're sort of smack in the middle, as you see here. And this allows us to really understand, well, what would happen if we do nothing at all? Would woodland naturally regenerate, and if so, over what kind of timescale, and what kind of diversity would exist in it? And it also gives us the ability to measure lift against that control. And again, the control is replicated 8 times across the site. In terms of the planting, we're studying 2 different types of forest. The first is a mix of broadleaf with species which would naturally occur in Wales and occur on the site. In fact, we have quite a lot of mature oak on the site, and oak is one of the ones that we're planting together with birch and rowan and aspen and wild cherry, giving us a broadleaf mix, which gives us the ability to really simulate what would have been here. If you went back 200 years or more on this site, you would have found a mixed broadleaf forest. These species are really chosen because they are probably primarily the pioneer species. These are the ones that would naturally come in first. And obviously what we're doing here is we're giving nature a helping hand to re-establish the broadleaf forest. But also within our scientific area, we're also planting some plots of conifer as well. And that's in particular so that we can compare broadleaf and conifer. This is important because the conifer really mirrors what happens in commercial forestry. And we cannot look at forestry and trees in general without thinking about what happens on the commercial side as well. And an awful lot of data comes about, in fact, from the commercial forestry industry. And by planting some cells which are commercial forestry style, we can then have a like for like kind of comparison as part of the test. We're also looking below ground, and this is where we begin to think about the fascinating symbiotic relationship between trees and mycorrhizal fungi. If you think about each of us as individuals, in our guts we have bacteria which help us as individuals, as humans, to process and digest our food and absorb key nutrients. In exactly the same way, trees need mycorrhizal fungi, which actually extend their root systems out by as much as 100 times to capture, break down and capture critical nutrients in the soil and feed them back to the trees. And the trees in return feed them sugars, which then help the mycorrhizal fungi to grow. It's an amazing symbiotic relationship that is, the more you dig into this, the more fascinating it is. So one of our treatments is to inject a scoop of soil from a forest, an established forest, it's a little cupful if you like, into the planting hole to try and jumpstart the mycorrhizal fungi on site. And the challenge here is that if you just go and plant trees in a piece of farmland that has been grazed for many, many years, for example, by sheep, then there will be nowhere near the richness of mycorrhizal fungi that you would find in a mature forest. In a mature forest, in a square meter, you might find as many as 60,000 different types of different forms of fungi and bacteria and etc. going on, all helping the process of breaking down things and storing that as carbon, storing carbon into the soil. So the panel is going to go into that in more detail, and they are real experts in that area. The second nature-based treatment we're applying is crushed basalt. And you can see here from an aerial that we took with a drone back in November timeframe, how we've spread this bluish, greenish, bluish, I guess, turquoise-coloured crushed rock on the planting areas. Crushed basalt is part of what's sometimes called enhanced rock weathering, and David Beerling is one of the world's experts. He will no doubt talk about that in some depth. And it is also a really hot technique right now, because when you apply the crushed basalt, it absorbs carbon dioxide from the air as part of a chemical reaction, chemical weathering, where as rainwater falls on the stone, it breaks down into calcium carbonate or limescale kind of stuff, and provides nutrients for the soil. So you can think of this as having two effects. One is it provides a vitamin shot to the new trees, and the second is that it's actually drawing down carbon dioxide from the air in quite significant quantities. There is also some intriguing research, which when I remember my first conversation with David, when we were talking about basalt addition, he said, you do realize, of course, that there is a potential interaction here between mycorrhizal fungi and crushed rock. And of course, that is quite intriguing, because the research suggests that the rock indeed can provide the nutrients, which helps the mycorrhizal fungi find the nutrients, which provides the nutrients to the trees, which provide sugars, which helps the mycorrhizal fungi to grow faster, to break down more rock. And obviously we have to test this. There's some research actually fairly recently from a group in Antwerp suggesting this needs testing at scale. So luckily here we are doing this. So the combination is really quite kind of intriguing. And over the course of the next 2 years, we'll be studying really tree survival and tree growth. We'll be using volunteers as part of a large-scale citizen science project to do this. So if you want to get involved, then we'd love to hear from you. And we believe that getting people involved in nature and in science is actually really important in general for all of our education, really, around environment and environmental topics. The team at Kew in particular will be looking at roots, and in fact they've got quite a large quantity of bare root trees which they're analysing at the moment, and looking at how the fungi have developed in the soil, and equally the fungi that came in on the trees as they came in and were planted originally. We'll also be measuring as well what the trees are drinking, so effectively the water, the soil pore water which is in the ground, using an instrument here which is called a lysimeter, which allows us to take a sample at root depth so that we can understand exactly what the trees are drinking. This is quite important in also allowing us to measure the carbon which is sequestered by the basalt as it breaks down. So when you put that all together, we have quite a complex picture, 72 different test cells. In total, 25,600 trees across 28 acres. So this is not a little bit of a corner of a field where we've planted 20 trees and we're comparing it with 20 other trees. We've got quite a lot of treatments, 8 different treatment types that we're looking at, plus control, and we're replicating that 8 times. And this really is a test grid which we can use and which scientists will be able to use for generations to come to test out different environmental topics. And of course, critical to all of that is the readings that are taken, the baseline measurements which were taken before the site was set up and are then stored, and the readings taken by volunteers and scientists over the years, over time, which provide a baseline and can then provide real data which can be used for climate modeling. So none of this is of course possible without our erudite panel. So at this point, I'm going to hand over to Colin, who's really one of the first to be involved. And Colin, I'm not sure if you remember, but when I first contacted you, I asked you what I thought was a relatively simple question, and you sort of rolled your eyes a little bit and educated me that, in fact, understanding what it takes to maximize carbon sequestration in trees and soil is not a simple question.
Yeah, I remember that. And I also remember telling you that setting up a field experiment is really hard. And so it's just so impressive to see it at that scale and step back and see the aerial photos and 25,000 trees. That's amazing. I can't believe we did it. I can't really believe you did it, Charles. It's impressive. We're excited. But yeah, Charles came to me probably a year and a half ago now, asking, this question about, how do we plant trees and think about reforestation in a way to, really maximize the carbon capture of these programs that are being rolled out all over the world. And I personally study the forest microbiome, these incredibly complex, fungal and bacterial communities in soil, particularly mycorrhizal fungi that grow on the roots of trees. And for the past few years, I've been leading a team at the Crowther lab at ETH Zürich, where, the wider group does a lot of work on reforestation. So, for a while, I've been thinking, how do we begin to inject what we've learned about sort of hyper diverse soil communities into our approaches to restoration and reforestation? And so when Charles approached me about this, I couldn't resist. I jumped at it and I started talking about these fungi and how, in general, when we plant trees to restore an ecosystem, we never think to plant, this forest microbiome, this bacterial and fungal community that we know is super important. And it's really remarkable to me because we know that in ex-agricultural landscapes, like the one we're working in Wales, and actually where most global restoration happens in these ex-agricultural landscapes, we know the soil communities look nothing like the bacterial and fungal communities that are living within intact forests. There's just completely different subsets of organisms living there below ground. And so I had seen some scientists recently publish on work sort of accelerating restoration in grassland ecosystems and shrubland ecosystems by essentially doing what's analogous in medical microbiology to a fecal transplant. They would source soil from a healthy, intact ecosystem, which is teeming with all those living fungi and bacteria and also soil animals and other things, and essentially use it as microbial inocula for the places they were trying to restore. And what they saw were absolutely huge effects. They could massively speed up restoration of those ecosystems. And so I just thought we need to do that, but we need to do it in a forest. And we need to not only quantify how quickly, these communities reassemble and the ability of, these microbial communities to enrich the biodiversity above ground, but also how those microbial communities might be able to affect the rate of carbon capture and carbon drawdown of the forest and the entire ecosystem. And so ultimately, the idea is that if we can take a more holistic and a more complete approach to restoration, considering both above and below ground components of biodiversity and of an ecosystem, can we create positive outcomes for both biodiversity and carbon removal at the same time? And so while I was doing that, I was actually already in conversation with Martin and Laura at Kew about their approaches to capturing and measuring below ground biodiversity, particularly mycorrhizal fungi. It's something they're really experts in. And so I approached them about how can we work together to sort of test some of these ideas in Wales. And so I think I'll hand it off to Martin and Laura here.
All right, thanks, Colin. So yeah, indeed, when Colin first contacted us, it was sort of trying to dive into the non-trivial issues of how to manipulate this below ground part of these future forests. And Colin and Charles did a good job of sort of laying out the overall design and setup here. And I want to pick on something that Charles said. He was talking about how you have the trees and then you have the soil, and that's sort of the key issue. In between the trees and the soil, the interface are these fungi, these mycorrhizal fungi. And it's a sort of a terrible word. It sounds like an insult, but it just means fungus root. It's something very ancient and incredibly common, but because it's in the soil, it's sort of out of sight, out of mind. And I don't want to neglect that there are some pretty big discovery issues to this trial. It's not just all figured out and there's a book that we can follow on how to optimize carbon sequestration using fungi. So it is, there is very much quite a bit of uncharted territory there. in this below ground. So I would even go as far as saying that trees don't have roots. They have mycorrhizas, and the trees don't interact with the soil or with the basalt or with other organisms. They do it indirectly through these fungi. So these people like Colin in recent times have been showing how on a global scale, different types of mycorrhizas, different combinations of fungi and plants have a really huge impact on the things that we care about: carbon, nitrogen, phosphorus cycling on a global scale, all the biogeochemical cycles that we are sort of messing with on a very mind-boggling scale currently. So in this particular trial, there is quite a range of different plant fungal combinations that we're potentially playing with and testing. So there are these sort of so-called endo mycorrhizas, or inside. These are the most ancient. And this would be on things like rowan or cherry, but also on the grasses that are already in the grassland. These have relatively fast carbon cycling, and they're generally adapted to low phosphorus soils, more or less. There are also ecto, or sort of outer mycorrhizas. These are on things like your spruce, your oak, your birch. These are more or less slower carbon cycling, and they're often associated with low nitrogen soils. And on top of that, we have some trees that have both, so the best of both worlds. So they have ecto and endo types, so things like poplar. So that already gives you an idea of just the many layers of complexity here nested within this very large experiment. And I think it is really exciting actually to be doing this on the ground and actually confront this complexity and trying to learn from it. So, in this day and age, we're all thinking about transmission, right? The microbial transmission is the thing, right? So how do you get COVID, and how do you not get COVID, and what do you have to do? So that is trying to avoid an antagonist, okay? We're trying to minimize transmission. Here, what we're trying to do is maximize the transmission of certain types of fungi, okay? So it's the complete opposite problem. What we're talking specifically about here is spores that are viable. So this is how fungi get around, how they sort of colonize new roots. And this can be moved around by, say, wind or soil or even animals, versus direct root-to-root contact, which is what you would have in a mature woodland, where you have mature trees that are established and then new seedlings that get directly colonized. So I kind of, now that we've laid out these various complexities, I think it's a good time to pass it on to Laura, who will tell us what we are doing at the moment to try to sort out these various interesting complexities.
Thank you, Martin. Hello, everyone. So at Kew Gardens, and as part of this project, what we are doing is to study the influence of mycorrhizas in carbon sequestration and storage across the different tree species that are being planted, and also across the treatments that they have been mentioned before. Using soil from nearby established woodlands with similar tree composition than the trees that are planted will accelerate the colonization of the roots by these mycorrhizal fungi that Martin has been talking about. And hopefully it will also speed up those beneficial functions that they have, not only for tree growth, also seedling survival, and all these functions that these fungi develop in soil, like carbon sequestration or nutrient cycling. So what we will do in this project is to analyze the mycorrhizas in the roots of the seedlings being planted in Wales, but also after experimental year 1 and experimental year 2. And we will assess the levels of colonization by these fungi of the roots of the seedlings, and also we will identify the diversity of fungi forming mycorrhizas. Depending on the type of mycorrhizas, depending on the tree species, as Martin mentioned, there are some that are ectomycorrhizas, other ones that are endomycorrhizas, we will have to use different approaches. So we will assess the level of colonization and the presence of these fungi in the roots through direct examination with microscopy. But in some cases, we will need to stain the fungal structures to be able to see them within the roots. And because we always look at roots, we identify the fungi in the roots, and many of these fungi don't show enough characters, morphological or anatomical characters, to be identified at the species level. So we will use DNA sequencing techniques to identify them, comparing the sequences that we obtain with reference fungal DNA datasets and bioinformatics. So back in December 2019, when we thought that the plantation was going to take place in 2020, we didn't know about COVID at all. We carry out the small analysis of a set of seedlings from Mellor Nursery. That is the nursery that is providing the seedlings. And we found that there are very few fungi in the roots of those seedlings, a handful per tree species, and also that there is a wide range of colonization levels. So we would find seedlings that weren't colonized at all and other seedlings that were highly colonized. So it is important to assess those levels of colonizations before. Now what we have, as Charles mentioned before, we have thousands of seedlings to look at. We are looking at a set of 100 to 200 individual seedlings per species that are being planted, and we will assess baseline levels of colonization and also the diversity of fungi in the roots. And we will compare this baseline data with the data that we will collect after year 1 and year 2. And this will allow us to test for the effect of the treatments over time. In terms of what we are expecting, well, we think that over time, we will see higher colonization levels in the roots. And we will also expect to have more number of species forming mycorrhizas as well. But in the case of the seedlings that are inoculated with soil from the established woodlands, we expect to see more diverse communities and also more close or more similar to those of the donor or forest sites. And we really expect to see that through changes in mycorrhizal communities, those both treatments, basalt and also soil inoculation, will increase carbon sequestration and storage in the plantation. So I think, well, finally, we really expect that our findings will help guide or inform similar reforestation practices across the UK. So I think it is time to me to introduce Bonnie. Bonnie, over to you if you would like to talk about soil carbon.
All right. Well, a theme, I think, of many of the previous speakers is that in terms of reforestation, soil is really where the magic happens in terms of accelerating carbon capture. So as Charles mentioned, in a mature forest in the UK, over 3/4 of the carbon in the ecosystem can be contained below ground. And we know from many studies across the world that planting trees in general tends to enhance soil carbon, but there is enormous variation in the size of this effect. And in fact, in some places and locations, planting trees can actually cause a loss of soil carbon. So it's really incumbent upon us to figure out why this variability and how can we bias our results towards rapid carbon gain under tree planting. So the other way that the soil really drives the response of the whole ecosystem to the tree planting efforts that we make is soil is where the nutrients are that fuel the growth of trees. And in fact, in this specific experiment, some of our major manipulations, the microbiome additions, and to some extent the basalt additions, the mechanism by which they work is enhancing nutrient supply to the trees that we plant. So we also want to keep a close eye on the nutrient cycles in the soil and look at the mechanism by which that enhances tree growth. So what we're doing in my lab is conducting a broad set of measurements, both to lay the foundation for future research at the site as we look at carbon gain over 10, 15, 20 years, and to measure parameters that we think are sort of the early indicators of the trajectory by which these forests will develop. So we're looking obviously at soil carbon. Now it can take many decades to see a change in bulk soil carbon, but we can also look at specific pools of soil carbon, classes of organic compounds in the soil that tend to change quite rapidly in response to the inputs of dead organic matter that the trees are providing. As Charles mentioned, we're also looking at the pore water of the soil, essentially the solution that trees drink. This is where we'll see signatures of the basalt addition treatment that David will mention in a minute. It's also going to give us some idea of what nutrients have been unlocked to fuel tree growth. And as a matter of fact, next week, a whole team of people from my lab are going out to look not only at the nutrients that are available in the soil, but what is in the tissues of the trees themselves. So by comparing these quantities, we can get an idea of how efficient the trees are at capitalizing on the nutrients that are held in the soil. And we expect this to vary as a function of both the microbial addition treatments and the basalt addition treatment. So again, one of the major potential breakthroughs that this experiment can provide is that we have an experimental platform to look at the long-term trajectory of carbon pools in the ecosystem, not only in the trees themselves, but below ground as well. And we are explicitly manipulating the parameters of the ecosystem that we think are important for these long-term carbon trajectories, the microbes that are there, the chemistry of the soil itself. And this really is going to give us unprecedented insight into how we can help other landowners guide their forests to maximum carbon uptake. And so since I've mentioned quite a bit about the basalt addition treatment and how that's going to interact with other soil parameters, I'd like to hand over to David Beerling, who can explain in more detail some of the other mechanisms by which this treatment can enhance ecosystem carbon capture.
Sure, thanks, Bonnie. So I should say I got involved in the project after Charles and Heather approached me. And I was really impressed by the goals, ambition, and the scale of the project. You saw from Charles's slides, I mean, the scale is hugely impressive. And I think Charles and Heather had heard of my work on enhanced weathering as a greenhouse gas removal technology. And this naturally led to us sort of discussing the idea that we could combine some kind of enhanced weathering treatment with afforestation and reforestation that they were planning. And really, you know, in the sort of scientific world, the jargon for this is co-deployment of two different greenhouse gas removal technologies. And really, this is a kind of Cinderella climate mitigation strategy. It's a research frontier, underdeveloped research frontier. But ultimately, it could provide a new sort of strategic route to strengthen and deliver the UK response to net zero, and also be translatable to other countries around the world. So just to sort of unpack the science a little bit, in case you're not familiar with enhanced weathering, surely not, then really enhanced weathering involves accelerating natural carbon cycle sequestration processes by amending soils with crushed calcium, magnesium, silicate rocks. And in our case, we're using basalt, which is a natural volcanic rock. And as this undergoes chemical reactions in the soil, it basically converts atmospheric carbon into inorganic forms that are then sequestered in soils. And in the context of the soil microbiome aspect you've been hearing about, the mycorrhizas, it turned out, just as Charles and Heather had contacted me, it turns out I had two grad students that were studying how mycorrhizas accelerate the weathering of basalt grains. And mycorrhizas have been mining nutrients for trees or plants out of basalt grains for 400 million years, ever since the first plants arrived on land. So they're pretty good at it. And it turns out that if you bury a bag of basalt in the soil of a pinetum, they're excellent at accelerating the breakdown of those minerals as they extract the nutrients to pass to the plants. And then in return, as the plants get more nutrients from the mycorrhizas, then the trees can provide more carbon to help the mycorrhizas proliferate. And so there's kind of mutualistic symbiosis going on here. And of course, as the weathering proceeds and releases these mineral nutrients, so the trees grow better and you have a higher tree productivity and you sequester more carbon into the wood, so you get better carbon sequestration. You can also expect some additional co-benefits from doing the basalt treatment, which is improved tree survival, because as you bury the basalt, it can stimulate the root growth and the mycorrhizal growth, improve nutrient uptake, and help the trees develop better and acquire more nutrients. And this has the benefit of maintaining tree density and reducing costs in terms of having to replant where your saplings have died. And so I guess the long and the short of this is really combining afforestation and reforestation with enhanced weathering has the potential to kind of increase carbon sequestration into forest biomass, improve your tree survival, while simultaneously delivering a kind of more cost-effective long-term CO2 removal. So it's very exciting to see this being co-deployed, and look forward to seeing how it develops over the next few years. I think that's all I had to say on that aspect of it. But Pete, over to you. Maybe you could tell us why you decided to join the Carbon Community's Scientific Advisory Board, and where, if you did a horizon scan, where does the Carbon Community go next?
Okay, so thanks very much, David. So I joined the Carbon Community because, as has been explained before, this is cutting-edge science. So it's the co-deployment of two techniques for which, each of which there's only a handful of experiments around the world on enhanced weathering and on mycorrhiza, mycorrhizal inoculation at field scale. So trying those things together is incredibly exciting. We think we know what will happen, right? We think the mycorrhiza will help the trees grow. We think that there will be increased carbon storage. We think that the enhanced weathering will draw down some CO2 and may also improve the productivity, but we don't know. Having been involved in science for 25 years, I know that science never goes the way that you expect it to. So I think what comes next is finding out what these interactions are and what the unexpected consequences and what the unexpected findings are, and I'm sure there's going to be some, and those in a way are just as exciting as confirming your expectations. So I think looking forward, that's something that we can do, is we continue to measure and we continue to look for interactions. We've been so far, we've been fairly focused on the carbon. I think moving forward, as we grow the community, we could get some people in to look at the biodiversity impacts, because we have to deal with the twin crises of biodiversity loss and climate change together. So I think that's something that we'll try to expand in more into the future. And also we could get more social scientists involved to look at the human well-being benefits of this particular forest and forest more generally. So I think we're just at the start of a few decade-long journey, and I can see this growing it already is something very exciting, but I can see it growing into something even more multidisciplinary and more interesting. But that's enough from me. I'm going to hand it back over to Charles.
Well, thanks, thanks, Pete. And you're quite right that the timescales here can be significant, not just because we're growing trees, but also because of this facility effectively that we're building for scientists to use and leverage. Is one of the key reasons actually that we're a charity, is so that, because of the way that charity law works, and the assets in the charity structure are locked in, and they will always be there even after we've shuffled off our mortal and there's some other team taking over. And actually, well, what a great international panel we have here. So thank you all panelists. I mean, we've got people from all over coming together on this, which is super exciting. If you have questions, then please put the questions in the Q&A box. That would be great. We've got some great questions already. So I'm going to start going through those, and I'm going to throw them over to the panel. So first off, let us start here with Amelia. Amelia, thank you very much for your question. It's a fascinating project. I'm a soil scientist from Swansea University. Brilliant, we need soil scientists. Great, go Amelia. Only 1 hour away from the site, and we'd be very keen to get involved in the project, possibly have undergraduate or master student projects on soil moisture, soil carbon, soil respiration. What are the opportunities for doing any studies at the site? So this is exactly why we built it. Exactly, it's there, it's instrumented, and we would love to bolt things on which are can build on what we're already doing. The fact you're local is brilliant. By all means, reach out to us afterwards. We're also working with Mary Gagan in the University of Geography at Swansea University, and in fact, she's already built some of the work that we've been doing, in particular around rock weathering, into some of the geography modules for undergraduates. So there's something there. But yes, we'd love to talk with you some more. Okay, so next question from Ian. So thank you, Ian, for your question. Amazing research with real implications. Well done. Did you consider adding biochar, say, up to, they just moved, up to 5% to the soil initially to enhance growth, or could it be part of future work? So I'm going to answer that, and then I'll throw it over the panel and get their views on biochar as well, because this will be an interesting one. It is one of the techniques we looked at, and obviously when you're doing a co-deployment trial, it starts mushrooming and gets big very quickly. If you want to replicate it 8 times, then all of a sudden the thing just gets bigger and bigger and bigger. So we did look at biochar, and actually we felt that the Basalt addition with as part of enhanced rock weathering showed great potential. But biochar is also interesting. We are having some early discussions with another group of scientists who are looking potentially again at some sort of a deployment on the site using biochar. So there is some opportunity there. But since we have this great panel here, so let us throw it over. Who would like to talk to biochar? Who wants to go first? Anybody? Go, Pete.
Yeah, just to say that it would be great to look at biochar. But as you said, when you had to look at the interactions between 2 factors, we're up to 72 plots. So if we have to look at the interactions between 3 factors, we're up into the hundreds of plots. So it would be impossible to do 3 together. But if you're going to do some more planting at the site, if there are other areas that you've yet to plant up, you could do a biochar, no biochar replicated trial, and I think that would be interesting in its own right. I think it's worth a look.
Anyone else want to jump in on biochar?
I guess I could just add, we are running at the moment in the Leverhulme Centre, we're running mesocosm experiments where we're growing crop plants with crushed basalt, and then with crushed basalt and biochar. It's an interesting combination. Excuse me, but we're not seeing huge additive effects of biochar on its own or biochar with basalt. And I think the literature is very sparse on looking at that interaction in the field. But as Pete said, it would be interesting to try it. But there's obviously logistical challenges in trying to maintain a manageable experiment at the field scale.
Okay.
I also think we were super interested in investigating some things that hadn't been explored nearly as much. There's actually, comparatively, I know there's still plenty of biochar research to do, but there's been a lot of investigation into biochar. People are starting to scale it up. There's like no shortage of startups out there applying biochar. We felt like these are 2 potentially really important aspects of natural carbon solutions that could really, move the needle and accelerate carbon capture that really weren't being investigated in a lot of other places. And so we thought these were great places to focus our energy.
Okay. All right. So let me jump to a question from Pete, a different Pete actually. Is the hypothesis that trees need good enough conditions to achieve their genetic potential and carbon sequestration, or that these approaches will boost production and sequestration beyond existing yields on good woodland soil? That's a lovely question. Who wants to jump in on that one?
I'll jump in, Charles. Yeah, I think the idea here is that we can sort of jumpstart what would naturally happen anyways in these ecosystems, right? Like we're moving mycorrhizas in, but if we left this site alone for hundreds of years, we suspect eventually all of these this assisted migration of microbes we've done would eventually catch up. They'd eventually show up. The real question is, how long does that take? And there's more and more evidence to suggest it takes a really substantial amount of time. And there's also a lot of fungi that don't show up in these forests until really much later stages. And so the idea that we can essentially just accelerate this process is what we're doing. So, you know, whether the soil is good or not, I guess in one sense, perhaps it's less quote unquote good because it's perhaps missing and depauperate of a lot of these organisms. But I'll let somebody else speak to the sort of enhanced weathering side of that question.
Is the hypothesis trees are good enough? Well, I think really what you want is just conditions that ensure tree survival, obviously. And the better the trees grow, the better they're going to weather. The healthier they are, the stronger their root systems, the more productive their mycorrhizal systems, the better they'll drive weathering and carbon sequestration. So good enough would be good, but better, you know, is going to get you even more carbon sequestration.
Yeah, I mean, let me give a viewpoint on this as well, because if you look at the site that we've chosen here, this is an upland site. It goes from about 150 meters above sea level at the bottom up to about 280. So 280 above sea level is getting quite high, and you certainly know when you're there. You feel the exposed conditions every now and again, you know, as the wind and wet drives through. But it's also typical of a lot of agricultural land as land use changes in the UK. A lot of sheep farming land that will ultimately come into forestry or be forested in one form or another. So this land had been grazed by sheep for many generations, and as a result, you know, that is what it is. So I think there's another dimension to this which is kind of interesting, which is this can be quite representative of what you might consider to be relatively poor agricultural land because it's upland. You know, you can't really use this for arable farming or something like that, which then could be forested, and therefore that the potential to then think about that more broadly across the UK is kind of really quite kind of interesting. In fact, that's a bit of a segue. I'm going to jump to Alain's question. How is the King Hordey site chosen? What is the underlying geology? And are the soils typical of Welsh brown earths? And another one from Swansea University. So go Swansea, you're rocking tonight. So the site was chosen for a whole combination of reasons. I'll give you sort of my take on it first, and then see if anyone on the panel wants to jump in. But we chose the site because if you look at great places in the UK to grow trees, then you could be looking at Scotland, and you could be looking at Wales in particular, because they are nice and wet, and obviously trees want wet and all the like. The land is also more affordable than if you try and do something, for example, the southeast, where it might be dry and super expensive. So the combination of those things are important. We also like the Welsh site actually because that access is good, and in particular if we wanted to build a scientific research facility where people can come and use that, we don't want to have to put everyone on an aeroplane each time, even though they could pop in and see Pete when they're up there. But you know, and as a result, you know, the carbon footprint goes up, and it's less accessible, and it's not as easy to go and take measurements and all those sorts of things. So access was also pretty important. So we wanted somewhere that sort of fit the bill there. It's also as a special site as well. It's very pretty, and therefore it's something that you know, hopefully people will want to go to and enjoy when they're there. As you saw from some of the photographs, this is a beautiful part of the world. It's not very well known in some ways, and yet it's a beautiful place that you can access through public transport. It's a whole combination of those things that chose the site. In terms of the soil, it's a typical a peaty clay. We have got some patches which are peat bog, where we obviously haven't planted trees, and where we will look in time to actually restore peat bogs, and we'll have peat bog ecosystems there as well on site, which is another sort of interesting dimension for us. Does anybody else want to jump in on the site? Bonnie, you've been there. Any observations, thoughts?
Well, I wouldn't recommend camping there overnight in a cold snap in April, but it is spectacularly gorgeous. And I think, as Charles said, one of the advantages of it is that it is typical of the land which would be available for afforestation throughout the rest of the UK. So the idea is not necessarily to choose the site where we already know the trees could grow the biggest or the strongest, or which has the richest soil, but to get a perspective on what would be the maximum carbon drawdown that we could achieve given the types of land that would be available for these types of projects at a national scale.
Okay, great. So another question, this one from Andrew. Really interesting presentations. I wonder what's the carbon footprint of the crushed basalt? So let me give my quick take, and then David, I'll throw this one over to you. So first off, we were very fortunate with this site. It's also only 20 miles away from a basalt quarry. As you may know, if you're a geologist, a lot of the basalt is on the western side of the UK. You don't get much in Kent. And we're very fortunate it's only 20 miles up the road, and therefore the actual transportation is reasonable. It's not so far. As you dig into the way that basalt is produced and what it's produced for, it's mainly produced for asphalt and used in roads in the UK. And the roads sort of specify that they want a certain size chip, that then goes into the asphalt. And as a result of producing those chips, you end up with a lot of dust, which is basically a byproduct. And they produce way more dust actually than can be sold, and there is a market for. So there is opportunity here basically to take a byproduct out of mining, which was going to get produced anyway, rather than something you have to mill specifically to then put on your fields. We will be doing more work on the actual carbon footprint of the basalt in this particular case, but we're not there yet in terms of being able to sort of talk about that in more detail. David, do you want to talk about in general the footprint on
Sure.
basalt?
I think that's a very nice summary. I mean, there've been a number of studies that have looked at carbon footprint associated with mining, grinding, and spreading the rock. And all of them show that enhanced weathering is net negative, but that the hotspot is the grinding of the material. And what we found in the UK is that actually, as Charles just said, there are a number of basalt quarries both in Wales and also in Northumberland. And they're mining it for aggregate, for road building and so on. And for every ton of rock, 20 or 30% of it produces this too fine a material that they can't use. And they don't have an opportunity to sell it. And so it's accumulated. And we were fortunate enough to have our Leverhulme work on the BBC News a couple of years ago. And after it, a guy from Cemex rang me up and said, David, we've got like 20,000 tons of basalt rock dust we don't know what to do with. And it's just stockpiling at our different quarries. Can you help us? Is it any use to you? And so suddenly we saw there's an opportunity here for taking a waste product and helping crops grow better, restoring soils, and capturing carbon. So there's an opportunity to build what's called a sort of circular economy. And of course, by doing that, you're avoiding that CO2 hotspot of grinding, because it's just a byproduct. And that same logic and rationale fits into this study as well.
So David, whilst you've still got your mic on, there's a related question to the basalt. Did you consider olivine for rock weathering, which is arguably more effective than basalt? So I think this is a story about heavy metals, isn't it?
Sure, yeah. So olivine does weather faster, but it does have sort of trace amounts of chromium and nickel, heavy metals that are quite nasty that you wouldn't want to put in the soils. There's also an issue with the supplies of it. Sometimes it comes from Hartsbergite mines or dunite mines, and some of these are associated with asbestos and have been closed down in recent years. So, all of these factors together, although you get more bang for your buck in terms of carbon capture because it weathers faster, I think you're safer using basalt, which is a multi-element product. So as it weathers, it gives this vitamin shot to the trees to improve carbon sequestration that I was talking about and Charles mentioned earlier. So yeah, all in all, we think basalt is a better bet than olivine.
And it's very widely available too, isn't it? I mean, it's one of the most common rocks on the planet, I think, isn't it?
It's one of the most common surface rocks out there. No shortage of basalt.
Cool. Okay, so we are rapidly coming towards the top of the hour, so we have a couple more questions. So this question from Alan. So thank you, Alan, for your question. So as a non-biologist measurement engineer, can I ask how will the experiment account for existing and changing microclimate effects, e.g. wind, sun, rain variations across the site, including groundwater flows, which will likely change significantly over the years and as tree roots become established? Right, so who would like to talk about measurement effects?
Happy to talk a little bit about it. So the biggest way we overcome sort of all the types of variation you're talking about, which are inherent to like anywhere you do a field experiment outside, wherever you are in the world, is that, this slope is sunnier than this slope. Perhaps there's below ground water flows we're not considering. And so the way we overcome that is through a block randomized design. So each of those test cells is actually embedded within a larger block that gets placed on the landscape, and then we randomly assign the treatments within there. And then by replicating those blocks many, many times, the idea is that we've created enough random variation such that even if there are big flows of water or pre-existing site conditions that we've missed, they're equally likely to be in each type of treatment, and we've replicated enough such that we can overcome that and still have the statistical power to see the main effects of our treatments, which are not randomly assigned, but very intentionally assigned. And so that's the biggest way we do it. But we're also measuring some of these things too, however, so we can account for aspect and slope and some of these things. And because we're measuring so many trees and we know their positions, we can also use spatial statistics to overcome some of this as well and sort of build a little bit more power into the experimental design.
Cool. Anyone else want to jump in on that one?
Anyone listening is a hydrologist or interested in microclimate effects and would like to get involved, please do, because the more things we measure, the more power we have to explain the patterns that we end up seeing.
There we go. Your call to action. Right, fantastic. There's one other question here, and then I think we're going to wrap up. So was there any consideration to use conifers in species mixes rather than just spruce? The site looks like it's suitable for other species like Douglas fir that could grow in mixture with spruce. There is work by Forest Research which talks to this. So thank you for the question. So when we were looking at tree species mix, we did look in particular at Douglas fir, and we were advised that the site is relatively high for Douglas fir, and yet bizarrely you actually see some mature Douglas fir very close by at the same altitude. But at 280 meters above sea level, it's getting towards the high end. Is what we were advised. And the second consideration is actually doing a mix within conifer. If you are putting in Sitka spruce, for example, alongside, say, Douglas fir, we were also advised that Sitka is a bit of a brute and doesn't play nicely with others. And as a result, it's one of the reasons why it gets planted in monoculture kind of plantations. And if you look at the most popular tree planted for commercial timber, it is Sitka spruce, and it's planted in great big blocks. And that's because it's been developed over many years, and it grows very fast and it grows very tall, and doesn't have many branches coming out to the sides, and therefore you end up with long straight bits of timber without too many knots in. So I think that we've ended up with little monoculture blocks, which are kind of broadly representative of the way that commercial forestry works, and therefore that's a reasonable basis for doing this kind of comparison I talked about side by side. Right, okay, so we have a minute or two. Could I just ask the panel, is there any part of this story that we haven't brought out today that you think as you'd like sort of a last parting sentence to say, yeah, we really didn't talk about this, and this is important, we should talk about that?
I think perhaps just to mention that, this is a long-term trial, emphasize that done the hard work setting it up, but the carbon sequestration is going to follow after several years of measurements and intensive campaigns.
Okay, it's a long-term gig. Anyone want to build on that?
Well, I guess it's sort of stating the obvious, but in understanding change, what we are really limited by is this sort of long-term kind of data that is, standardized. And this is one of those opportunities to actually be there when you start to set it all up, so that you actually collect the data that we as scientists would want.
Yeah, that's a great point. In fact, quite a few people have told me that the number of forest research sites where you could research a forest in the UK has dwindled over the years, and most of the existing ones, there's only a handful, maybe literally 5 or so others, are mature forests rather than something new. So the ability to take measurements and instrument the thing from the ground up, representing new forest creation or restoration, is really, really important. So, Colin, did you have one? I saw your mic came off for a moment.
No, I was just going to agree with everyone, especially about the time series. Like for soil carbon and whole ecosystem carbon balance in general, it's rare to have sort of these repeated measures through time. And those are the things that help us understand how well we can predict dynamics and how these things are changing in time. Most soil carbon analysis is just capturing giant variation in space, but not actually looking at these temporal dynamics. And that's because we're fundamentally limited by the measurements and the experiments that are available. And so that's why I think experiments like this with like really rigorous baseline data are super valuable. Okay,
great. Well, on that note, and we are a minute over, my apologies for being slightly late, but by way of closing, so first of all, thank you to all of our panelists. You've been a wonderful panel. This has been fascinating, and you've educated me, and I'm sure you've educated everybody else, and it's been great to hear from all of you. So thank you really for all of your time and effort on this. It's wonderful. Thank you also to the audience with some great questions. There are some follow-up actions there. People want to get involved. Bonnie's looking for help as well, by the looks of things as well. So please do jump in and contact us, info@carboncommunity.org. If you want to get involved in some way, we'd love you to get involved, either as a scientist if you've got some ideas, or as a citizen scientist if you just want to get involved and do some tree measurements with us and everything else and help contribute to the project. For more information in general, obviously ping us at info@carboncommunity.org, but you can also subscribe. We have a newsletter that goes out fairly regularly, and that's probably the best way to keep up to date with what we're doing. You'll find the box on our website at carboncommunity.org. And equally, you can also find out and keep track of us on social, and our handle at Twitter and Insta. Twitter is probably our main channel, is the Carbon Com. So please do jump in there and follow us. So without any more ado, and thank you to everybody from the Carbon community, let me just sign off and just sort of say, we're here. Our mission is basically for trees, for soil, for science, and for future generations. Thank you very much.